All right, continuing with cytoskeleton functions, stability, movement of the cell. Earlier, we talked about how a cell can move, in particular like a white blood cell that's going to engulf a bacterium, like a E. coli cell, through phagocytosis. Well, the movement of the plasma membrane to form pseudopodia to engulf a plasma membrane, that's movement. And what's moving the plasma membrane, the pseudopodia, is components of the cytoskeleton that are pushing the plasma membrane out. Engulf something, that phagocytic vesicles move into the cell. Well, that movement of the vesicle into the cell is again through the action of the cytoskeleton. Cilia and flagella, which are longer extensions of the plasma membrane, have cytoskeleton components within them, and they move, thus moving the cilia and the flagella. And we'll see that later on as well. So non-membranous organelles, centrioles within a centrosome. So a centrosome has two centrioles in them, and they are organizing structures that will move chromosomes during cell division like mitosis, as we'll see later. Here's our cilia, a long extension of, well, a relatively long extension of the plasma membrane that if the cilia move and the cell is stationary, it allows material to move across the surface of the cell or across the surface of a tissue if the cells are ciliated. Okay? So within the cilium, which is single for cilia, there's components of the plasma membrane that allow movement of material. Flagella is essentially a very long cilium. Structurally, internally, they're very similar. The difference is that they're very long, and usually cells that are flagellated are mobile, and the only human flagellated cells are sperm cells that swim through the woman's reproductive tract. Here's a picture of a ciliated single-celled organism that has cilia on it. So continuing with our non-membranous organelles, ribosomes, the site of protein synthesis. There's two subunits to each ribosome. Don't worry about that. But a informational molecule, something called mRNA, will latch into a ribosome, and the ribosome will bring in amino acids to synthesize, produce a protein. So ribosomes are the sites, the structures where proteins are produced. There are two types: free ribosomes that just are within the cytoplasm of the cell, floating around basically, and they produce proteins that are destined to stay within the cell, within the cytoplasm, to do functioning within the cell. Attached ribosomes that are on what's called the rough endoplasmic reticulum, the rough ER, they produce cells for export. Those cells get packaged into a transport or secretory vesicle, and that vesicle latches onto the plasma membrane, fuses with the plasma membrane, and that material gets ejected through that process called exocytosis. The mitochondria. Mitochondrium is singular. They actually have a double membrane, excuse me, and this is the site of production of ATP, that high energy molecule. So this is a shorthand equation for a sugar molecule like glucose. We breathe in oxygen at the mitochondria. Here's our organic molecules like carbohydrates, glucose, and oxygen. The mitochondria use an elaborate series of metabolic chemical reactions to produce ATP and our waste gas, carbon dioxide. So cells that have a high energy ATP requirement, like muscle cells, have many mitochondria per each muscle cell. Has an inner and outer membrane. The nucleus, housing the DNA, the genetic material within the cell, so often called the control center of the cell, double membrane, and there's nuclear pores that allow material to move into and out of the nucleus. DNA within the nucleus has two configurations. It's either loosely associated in the nucleus, and in that condition, the DNA is called chromatin. When the cell is getting ready for cell division, whether it's mitosis or meiosis, the DNA gets replicated or duplicated, the DNA gets tightly condensed, bound up into structures called chromosomes. So when the cells are undergoing cell division, the DNA in the form of chromosomes usually is readily visible. And then the nucleolus, this little kind of dark region here within the nucleus, that's where ribosome synthesis occurs. So the genetic material for production of ribosomes occurs within the nucleus at the nucleolus. Ribosomes exit the nuclear pore, and then the ribosomes are either free or attached ribosomes in the cytoplasm. The ER, endoplasmic reticulum, elaborate network of membranes and cisternae, which are like outpocketings, of the membrane, like cavities in a will, enlargements. 4 basic functions. Synthesis of things like proteins that we talked about, but also lipids and carbohydrates. Storage of molecules that are synthesized and or absorbed by a cell. Transport. So molecules will move within the ER, endoplasmic reticulum, because it's fairly large and elaborate. And also detoxification of drugs and toxins. So for example, our liver cells have a high amount of endoplasmic reticulum because one function of our liver cells is to detoxify harmful substances. So here's 2 types of ER, endoplasmic reticulum, rough and smooth. The rough ER has ribosomes attached to the surface, giving it a bumpy appearance, hence the name rough ER. And if you remember, ribosomes are important in protein synthesis. So one main function of the rough ER is protein synthesis. The smooth ER, over here on your right, is called the smooth ER because it has a smooth membrane appearance. Since there's no ribosomes, it's not involved in protein synthesis, but it synthesizes things like lipids, steroids, and carbohydrates, stores calcium, as we'll see when we do muscle contraction, muscle tissue, and then detoxification of harmful substance is another function of the smooth ER. The Golgi apparatus, kind of like this elaborate membranous structure, multiple layers, stacks of membranes in the Golgi apparatus, and it's involved in protein synthesis and export. Also packaging of enzymes that would be packaged within a vesicle, and then the vesicle is then a lysosome floating around in the cell with digestive enzymes to destroy things like bacteria that might be engulfed through phagocytosis. Packaging of materials to be secreted out of the cell like proteins. Proteins produced by the rough ER would get put into a vesicle. The vesicle would reach the Golgi apparatus. The proteins within the vesicle, now in the Golgi apparatus, would migrate through the Golgi apparatus, be modified, edited, put into a transport excretory vesicle, and then that vesicle would fuse with the plasma membrane, and then those proteins would be secreted or ejected through exocytosis. Oh, here we go. So here's the rough ER, ribosomes, these little red beads are amino acids forming a polypeptide that becomes a protein, put into a transport vesicle, that's moved to the interface or the cis face of the Golgi apparatus. Protein migrates through, edited, processed. The protein then gets put into a vesicle that can become a lysosome to do its functions within the cell, or the protein can be secreted via exocytosis, or the protein, if it's destined for the plasma membrane, will then be inserted into the plasma membrane as that vesicle fuses with the plasma membrane. If you remember when we looked at the plasma membrane, there was a number of embedded proteins in the plasma membrane. How did they get there? Via a transport vesicle from the Golgi apparatus. Oh, and the movement of all these things, the transport vesicles and the secretory vesicles and the lysosome, all that movement, if you remember what generates movement, cytoskeleton. Those lysosomes, here's another diagram of the nucleus, rough ER, smooth ER. Proteins get into transport vesicle, winding up with the Golgi apparatus or complex. Some of those proteins are digestive enzymes put into lysosomes. The lysosomes function to destroy things like bacteria and or even damaged, diseased, and or old cells. So some of the lysosomes will function to destroy the cell in what's called autolysis, which is a naturally occurring event. Peroxisomes, I believe they are, here we go, peroxisomes. They are similar in the sense of how they're formed like the lysosomes. Digestive enzymes are packaged into vesicles that become peroxisomes, and their main functions, the peroxisomes, are to break down organic molecules, and or proteins. Peroxisomes also contain an enzyme called catalase. Many enzymes end in ase, so catalase that functions to break down harmful substances like hydrogen peroxide. That stuff that you put on cuts to disinfect cuts. But hydrogen peroxide can be produced by certain metabolic reactions in the cell, and then that hydrogen peroxide would be harmful to the cell. So one function of the peroxisomes is to degrade, break down hydrogen peroxide to generate literally just harmless water and oxygen. Here's a diagram of the models that we have in lab, and you'll see this model in our lab supplement when we're looking at cells. And in the lab supplement, they'll be labeled like lysosomes are in yellow, peroxisomes tend to be purple or dark gray peroxisomes. See the bumpy surface here, that's the rough ER. Smooth surface over here, smooth ER, there's the nucleus. Golgi body or apparatus. So a general model in lab of a typical animal cell. Okay, let's look at intracellular attachments, how cells are linked to each other, attached to each other. There are cell junctions. There are gap junctions because there's a channel pathway between cells, gaps. And they're formed by these rivet-like structures with a pathway through them that are called connexons. And the connexons are made up of proteins called connexin proteins. And they allow coordinated functions between cells because they allow communications between cells. And we'll see this when we do cardiac muscle contraction of cardiac cells and smooth muscle contractions and within other tissues that have gap junctions. We'll focus on the gap junctions when we look at heart contraction, cardiac cell contractions. So what happens there is ions, which allow the electrical signal to move rapidly between adjacent cardiac muscle cells because of the function of the gap junctions. And that's what we see. Where are gap junctions? Oh, right down here. Okay? So that's one type of cell junction between cells, okay? Tight junctions kind of do what the name implies. Produces nice tight junctions between adjoining cells. So this in light blue is a plasma membrane of one cell. This is its neighboring cell. Tight junctions generate an impermeable barrier or an impermeable junction between cells so that material cannot sneak between cells. And that's important because if cells are involved in transport of materials across the cells to get into, say, the bloodstream in your small intestine cells are very careful, or one function of the cells in the small intestine is preventing material from simply slipping between the cells. And one way to prevent that is by the many, many, many tight junctions that tightly adhere or bind the plasma membranes of adjoining cells together. Tight junctions are tight. Desmosomes are really strong junctions, and what they do is they prevent adjoining cells from getting easily ripped apart, basically. So desmosomes are junctions